Good day, everyone! Welcome to another screencast in microbiology. I am your professor for today, Dr. Supakshay Basit, and for today's session, we will be talking about the microbial growth. So when we speak of microbial growth is actually the increase in the number of cells, but not on the cell size itself. So there are many factors that may affect the optimum growth of the organisms. So first, let's talk about the temperature. So temperature is considered to be as the physical requirements, okay? So because there are two main requirements, the physical and then the chemical requirements. So let's talk about the temperature. There are several terms that we need to define. So when we say minimum temperature, it is the lowest temperature that can support the growth of the organism. So when we say maximum temperature refers to the highest temperature that can be tolerated by the organism. And then when we say optimum growth temperature, it is the most suitable temperature, okay, that can optimize the growth of the organism. Okay? So therefore, having said that, we can actually classify organisms as two: psychrophiles, psychrotrophs, mesophiles, thermophiles, and hyperthermophiles. So if we're going to classify them, psychrophiles can actually grow, okay, at -5 degrees centigrade. Okay, so psychrophiles are actually can actually be found in embedded in the snow. Okay, whereas psychrotrophs can actually grow at refrigerator temperature, 0 degrees centigrade. Okay, mesophiles, if you will be looking at the temperature of mesophiles, you would notice that the optimum temperature is our body temperature. So majority of the pathogenic microorganisms are actually mesophiles. Okay, now if you will be looking at thermophiles are heat-loving organisms. So optimum temperature is 60 degrees, and hyperthermophiles, probably these belong to, if you remember, the Accdg, wherein they can tolerate a temperature as high as 110 degrees centigrade. So psychrotrophs are cold-loving, and they are the one that can cause food spoilage. That is the reason why they are actually very crucial for food industry. And whenever you are preparing food in the kitchen, so you do not want your food to be contaminated with psychrophiles or psychrotrophs. Then for the psychrotrophs, you would notice that the temperature in this range destroys most microbes, although lower temperature takes more time. So at this temperature, 60 degrees, it's a very slow bacterial cell. But this is the danger zone. It becomes lukewarm because there is a rapid growth of bacteria, and some may even produce toxins. So here, at this temperature, many bacteria will survive, some may grow. So this is already the refrigerator temperature. It will retard the growth of the bacteria. But those bacteria that can cause spoilage bacteria may somehow survive, but very few pathogens survive at this point. So now, if there is a deep freezing, there is no significant growth below the freezing point. Now, incidentally, some mesophiles may be considered as thermoduric, which means that they can endure high temperature because of the presence of the endospores. So again, we all know that endospores are actually made up of calcium dipicolinate that allows them to survive at high temperature for limited times. So Clostridium and Bacillus are example of genera, okay, that have endospores. Okay, now thermophiles are heat-loving bacteria because they can actually grow at temperature higher than 45 degrees centigrade. Okay, let's talk about pH. So most bacteria will grow between the pH of 6.5 to 7.5. Fungi such as molds and yeast will grow between 5 and 6, whereas Acidophiles can grow in acidic environments such as Helicobacter pylori, which can even survive the acidity of your gastric juice. So, microbes, okay, may grow over almost the whole pH range. However, the internal pH of most organisms is kept in the neutral range by either expelling the protons, the H positive, or the hydroxyl ions, the OH. So acidophiles are actually acid-loving bacteria that grows optimally between pH 1 and 5. So example, as what I've told you, is Helicobacter pylori, because this is the one that can cause duodenal, or no, that this can actually cause gastric ulcer. Okay, so bacteria found in acid mine tailings, acidic hot springs, molds and yeast, in pickled food, or even the old bottle of vinegar may contain acidophilic bacteria. Neutrophiles will grow between pH 5.5 and 8. So most of the aquatic, terrestrial, and mammalian sites would have this particular type of bacteria. So neutrophilic organisms that inhabit and infect humans have an optimum growth pH of 6.5. And the alkalophiles, okay, or the basophiles in some cases, okay, they grow optimally in the alkaline pH between 8.5 to 11.5. So an example of this, Vibrio cholerae, because Vibrio cholerae will need a pH of about 8.6 in order for them to grow. And some bacteria found in alkaline springs and ammonia-rich soil. Okay, osmotic pressure. So, if you're going to suspend bacteria in hypertonic environment, wherein you'll be increasing the salt or sugar concentration, this will cause plasmolysis. Okay? Those bacteria that can survive in high salt concentration, okay, we call them the extreme or obligate halophiles because they would require high osmotic pressure. And there are even some facultative halophiles that can tolerate high osmotic pressure. So these are the two possible scenarios that could happen. Here is an example of a normal isotonic solution, the normal saline solution, wherein the concentration of the sodium chloride is just 0.85%. So under these conditions, the osmotic pressure in the cell is equivalent to a solute concentration. So there's nothing changes when it comes to the morphology of the bacterial cell. Whereas, if you are to suspend bacteria in a high salt concentration, the bacterial cell will plasmolyze. What does it mean? It will actually shrink. If the concentration of solutes such as sodium chloride is higher in the surrounding medium than in the cell. So the environment is said to be hypertonic. So water tends to leave the cell, the growth of the cell is inhibited. So we call it plasmolysis. Now what will happen if you are to suspend the bacterial cell in a hypotonic solution? If that is the case, there would be plasmoptosis. So plasmoptosis is the eventual swelling and lysis of the bacterial cell. So water will come inside the bacterial cell. So there would be lysis of its cell. Okay, now let's talk about the energy and carbon source. So we can classify bacteria as two photoautotrophs. The energy source is sunlight and CO2 is the most common carbon source. Photoheterotrophs. The energy source is sunlight and the carbon source is an organic compound. Chemolithotrophs. Energy source could be inorganic compound and the carbon source is as CO2. And chemoorganotrophs, the energy source and carbon source are both organic compounds. So chemoorganotrophs could be parasite because they require living organic compound, or saprophytes. So when you say saprophytes, these are microorganisms that live on decaying organic matter. An example of a saprophyte is a fungus. Okay, so why carbon is important? Because carbon is one of the chemical requirements. It is a structural organic molecule considered to be as the energy source. So chemoheterotrophs would use organic carbon sources, while autotrophs would simply use carbon dioxide. So other chemical requirements would include nitrogen in amino acids and protein. Most bacteria can decompose proteins. Some bacteria may even utilize ammonium and nitrate. So a few bacteria will use nitrogen in nitrogen fixation. So these are the nitrogen fixing bacteria, and one of the manifestations that you're able to isolate them is when you found nodules in most of the roots of the plant. So there you can find nitrogen fixing bacteria. Now sulfur is actually found in amino acids, thiamine and biotin. So most bacteria can also decompose protein, and some bacteria will use sulfate or hydrogen sulfide. And phosphorus is actually needed in for the formation of DNA, RNA, ATP, and of course the membrane of the bacteria. So phosphate is an excellent source of phosphorus. Trace elements are inorganic elements required in small amount. So most of these trace elements would serve as the enzyme cofactor. So let's talk about the oxygen requirement. So we can classify bacteria as to obligate aerobe, facultative anaerobe, obligate anaerobe, aerotolerant anaerobe, and the microaerophiles. So if you are wondering what this culture medium that can support all of these type of microorganisms, this is an example of a thioglycolate broth. Okay, so let's talk about obligate aerobes. Okay, obligate aerobes are those organisms that would strictly require the presence of oxygen for growth. Without oxygen, they will not be able to grow. So an example of obligate aerobe is Microbacterium tuberculosis, because they reside in the alveoli of the lungs, which is very rich in oxygen. Now, facultative anaerobe is fundamentally aerobic by nature, but can still survive even in the absence of oxygen. So that made them flexible. So majority of the bacteria are actually facultative anaerobe, such as E. coli. Obligate anaerobe are those bacteria that cannot survive even in a very minute or small amount of oxygen, because for them, oxygen is toxic. So later on, we will be discussing what are the different toxic forms of oxygen. Okay, so this particular obligate anaerobes lack the enzyme system that could convert toxic form of oxygen into a non-toxic form. So what are these enzyme systems that they do not have? They do not have the superoxide dismutase. This is the SOD, and they do not have the catalase. Okay. Aerotolerant anaerobe is fundamentally anaerobic by nature, but can grow even in the presence of oxygen. Okay. In some texts, we call it facultative aerobic. And lastly, we have the microaerophilic. So when we say microaerophilic, these organisms can grow even in a very small amount of oxygen. Okay, so these are the toxic forms of oxygen. So these are the three enzyme systems that are needed in order for us to convert in order for the bacteria to convert toxic form of oxygen into a non-toxic form. So first is the singlet oxygen. It is the oxygen boosted to high energy state. They don't like that. And then the superoxide free radicals. Okay, so those bacteria that are aerobic are able to convert it to hydrogen peroxide in the presence of superoxide dismutase. So that's the first requirement. And then the peroxide anion should be converted using the catalase enzyme and peroxidase enzyme. So catalase will convert hydrogen peroxide into 2 molecules of water plus oxygen. So there's still an oxygen. And another enzyme system will convert this end product, okay, using peroxidase into 2 molecules of water. And then the last toxic form is the hydroxyl radical. Okay, so anaerobic bacteria do not have these three enzyme systems, the superoxide dismutase, catalase, and peroxidase. So even without oxygen, some anaerobic bacteria can still grow. As with aerobic organisms, anaerobe must obtain their energy from oxidation reductions. However, they can generate energy through fermentation reaction, which is the anaerobic breakdown of carbohydrates, or anaerobic respiration pathways, which utilizes inorganic electron acceptors such as nitrate and sulfate in place of oxygen as a terminal electron acceptor, like the one that is shown here in this equation. So in this equation, nitrate is reduced to nitrite by means of the enzyme nitrate reductase. So again, anaerobic organisms, these organisms will not grow in the presence of oxygen. So they deprive energy from fermentation and anaerobic respiration. So what are examples of anaerobic organisms? So we have the Clostridium species, the Bacteroides, Prevotella, and Porphyromonas species. Okay? So facultative anaerobe, these are fundamentally aerobic by nature, fundamentally anaerobic by nature, but can still grow even in the presence of oxygen. So these organisms grow well in the presence or absence of oxygen. So they are actually more flexible. If oxygen is present, they will often grow better as they make ATP through electron transfer chain to generate more than substrate level phosphorylation or anaerobic respiration. So majority of the Enterobacteriaceae such as Escherichia coli are classified as facultative aerobic. Aerotolerant means that these organisms, okay, grow in the presence of air but do not use it for respiration or energy production. So aerotolerant are actually anaerobic by nature but can still grow even in the presence of oxygen. So Streptococcus pyogenes is an example of this one, the agent that can cause sore throat. Okay, so this is an example of aerotolerant organisms. And lastly, we have the microaerophilic. They require a small amount of oxygen. Saturation of oxygen are actually inhibitory for their growth because they would only require about 2 to 10%, but higher concentration would become toxic for them. So the atmosphere is about 20% oxygen, so they do not like the atmosphere. So Treponema pallidum, the one that can cause syphilis, is an example of microaerophilic organisms. Okay, so let's talk about the organic growth factors. Okay, so the organic growth factors are organic compounds obtained from the environment. So this would include vitamins, amino acids, purines, and pyrimidines. So incidentally, purines and pyrimidines are actually nucleotide bases that make up the bacterial DNA. So macronutrients are actually acquired in the form of CO2, organic compounds, nitrates, ammonia, sulfates, and phosphates. The reason why we call them macro because they are actually supplied and needed in large amount. Unlike micronutrients, even the small amounts of these elements can actually be satisfied. The requirement for these elements can be satisfied by their presence in tap water. That is the reason why whenever you are preparing culture media, you have to use distilled water because you do not want micronutrients in the tap water to contaminate your culture media preparation. So speaking of culture medium or culture media are actually made up of nutrients prepared for microbial growth. So it is important that it has to be sterile. No living microbes so when you prepare. So autoclaving is important. Inoculum is the introduction of microbes into the medium. And once these microbes have already grown, so we call it culture. So culture are actually microbes growing in and on culture medium. So the reason why culture medium appears like a gelatin because it is made up of agar. So agar actually is a substance that can solidify culture medium in the Petri plates, slants if they are actually slant, and then dips. Dips are also known as the bath. So generally not metabolized by microbes. Like your gelatin, agar liquefies at 100 degrees and solidifies at a temperature approximately 40 degrees centigrade. So culture medium plural, culture medium singular, okay, they are chemically defined, which means that you know the exact chemical composition. And they are also complex because it is made up of even the extract and digest of yeast, meat, and plants, such as the nutrient broth and the nutrient agar. So if you'll be looking at the chemically defined medium, so these are the ingredients of the glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and water. For a chemically complex medium, we have peptone, beef extract, sodium chloride, agar, and water. So, you know, it's quite challenging to culture anaerobic bacteria because 20% there's about 20% oxygen in the atmosphere. So it's quite difficult to culture them. So that's the reason why we need to have a reducing media such as the thioglycolate or oxyrase that can combine oxygen. So if you're using, I told you a while ago that you can utilize thioglycolate in cultivating both aerobic and anaerobic bacteria. But if you are to use thioglycolate medium, it is very important that you heat the tube so that it will drive off the oxygen. So anaerobic medium actually uses the PRAS media, pre-reduced anaerobic sterile medium. Okay, so this system contains media which has been boiled to remove dissolved oxygen and then sterilized. So any air in the container is replaced with an oxygen-free gas mixture such as hydrogen, CO2, and nitrogen. So we can also utilize anaerobic jar. So in anaerobic jar, we place the plate inside the jar and then we open the packet that contain CO2 and hydrogen. So envelope containing sodium bicarbonate and sodium borohydride. So there is also an anaerobic indicator, which is methylene blue. So whenever there is a change in color, it means that there is a breach of oxygen. Okay, so catalyst pellets are also found here. So the one that I've shown you a while ago, this is the actual picture. So the most common method for incubating culture is either in anaerobic jar or bag, where the oxygen is displaced with a mixture of gas. So this can be accomplished either through a chemical reaction that results in an environment of hydrogen and CO2 gases through removal of oxygen by vacuum, and then we eventually replace it with a gas mixture containing nitrogen about 80 to 90%, hydrogen about 5 to 10%, and CO2 about 5 to 10%. So this is an example of an ideal anaerobic chamber wherein you have glove ports and airlock so that whenever you are processing specimen, it is ensured that there's no oxygen inside, because once there is an oxygen that contaminates your culture, organisms will not be able to grow. So the anaerobic chamber or glove box is used to inoculate culture media and for manipulation of culture. It is a closed chamber containing a gas such as nitrogen, which can displace oxygen. So we have another group of bacteria that are known as capnophiles or capnophilic. So capnophilic are organisms that require increased carbon dioxide for growth. So examples are Neisseria gonorrhoeae and Haemophilus influenzae. So the question is, how can you provide more CO2 in vitro? So we can actually use candle jar. So where the candle flames and consumes oxygen and would release CO2. Or we can use a calcium carbonate tablet. And as what I told you, this may actually utilize or an alternative to candle jar is the utilization of calcium carbonate tablets with water in order to release the CO2. So here's an example of the candle jar. Okay, so we lit the candle and then we cover the lid we replace the lid. And then, of course, if you replace the lid, there's no oxygen inside, and then the fire will burn out, releasing CO2. So it's a crude but cheap way of doing it. Okay? Whereas CO2 is actually more accurate because the calcium carbonate, okay, will actually release a more sufficient amount of CO2. So that's the CO2 part. So when you say selective medium will allow the growth of desirable organisms while inhibiting the growth of unwanted microorganisms. So selective media becomes selective by adding inhibitory substances. So differential medium, on the other hand, will allow us to distinguish colonies of different microbes. We can say that the microorganism is gram-positive or gram-negative. Okay, we can say that they are let me correct myself. We can see whether the organisms are lactose fermenters or non-lactose fermenters. So an example of that is McConkey agar. So incidentally, McConkey agar is also selective and differential at the same time. Selective because it allows the growth of gram-negative bacteria while inhibiting the growth of gram-positive bacteria. And it is also differential because it allows us to determine whether the organisms are lactose fermenters or non-lactose fermenters. So we could say that they are lactose fermenters if they will have pink or violet colonies, non-lactose fermenters if the colonies remain white. Enrichment media will encourage the growth of desired microbes. So let's say how to utilize it. So assume a soil sample contains very few phenol-degrading bacteria and thousands of other bacteria. So you do not want the thousands of other bacteria to overpower the phenol-degrading bacteria. So what we'll do is that we inoculate phenol-containing culture medium with soil and incubate, transfer 1 ml into another class of phenol medium and incubate, transfer to another class of the phenol medium and incubate. Therefore, so slowly but surely you are preventing other thousands of other bacteria to grow, and only phenol metabolizing bacteria will grow. So we can actually do that for salmonella. In salmonella, we are using selenite broth. For example, in stool, only few salmonella are found in the stool, but there are more E. coli. We do not want E. coli to overpower salmonella, so we place it in selenite broth. So culture it, okay, every now and then, and there will come a time that you eliminate unwanted organisms and you allow salmonella to proliferate in selenite broth. So a pure culture contains only one species of a strain. So a colony is a population of cells arising from a single cell or spore or form a group of attached cells. So a colony is often called colony forming unit or CFU. So how do we place or how do we inoculate our inoculum into Petri dish? We use the multiple interrupted technique in the streak plate. So as you can see, there are 3 streaks, and the last streak will allow you to have the isolated colonies. So hopefully in the laboratory, you'll be able to learn the actual way of streaking, okay, microorganisms into the culture media. So are you all excited to have microbiology or bacteriology lab? Well, your excitement is actually understandable because you will actually learn this technique if you're doing it, okay, rather than watching or having a virtual lab. Okay, preserving bacterial culture is possible by means of deep freezing between -50 to -95 degrees centigrade, and then lyophilization. We can do freeze drying, wherein the temperature is between -54 to -72 degrees, and then dehydrated in the vacuum. So let's talk about reproduction in prokaryotes. So usually, binary fission is the reproduction in prokaryotes. Yeast is not a prokaryote, and they may divide by means of budding. Fungi may divide by means of conidiospores and fragmentation of filaments. But for bacteria, it is the binary fission. So in the binary fission, the first step, as you can see here, the cell elongates and the DNA is replicated. So elongation of the cell. And then cell wall will have a septum. So you'd know that it's a septum because the cell wall and plasma membrane begins to grow inward, separating the DNA that had already replicated. And then the cross wall will form around the divided DNA, and then there is a cell separation. So here is an example of an electron micrograph showing a partially formed cross wall. So this is how bacteria actually multiplies. Bacterial cell multiplies. So do you know that each generation, there is a logarithmic increase in the number of cells. Okay? So if 100 cells growing for 5 hours produce 1,720,320 cells, so it means that in generation time, it will take 21 minutes, okay, in order for the bacterial cell to generate. So I will be attaching an animation on bacterial growth in this particular module. So generation time is the time required for one cell to become 2. And because of the generation time, we can actually plot the growth curve of the bacteria. So here is a graph that shows the generation time, wherein, as you can see, each generation, the number of bacterial population doubles. And this is the bacterial growth curve. We have the lag phase, the logarithmic or the exponential phase, the stationary phase, the death or the logarithmic or the decline phase. In the lag phase, this happens when you introduce bacterial cells into the culture for the first time. So here, bacteria will still try to adjust into their new environment and they will not grow. And then there would be an exponential increase, okay, during the logarithmic phase. This is the best time to perform the antibiotic susceptibility testing during the logarithmic phase. Here, this is likened to this graph, a logarithmic increase in the number of bacterial cells until such time that they will reach a plateau in the stationary phase. Why is it a plateau phase? Because the number of living cells, okay, is equivalent to the number of dead cells. Why do you think bacterial cell will start to die? Because here, exhaustion of nutrients may take place. In other words, the accumulation of metabolic waste product, exhaustion of the nutrients are the reason why bacteria had actually started dying here in stationary phase. Until such time that the population of bacterial cells decreases logarithmically as well. So we are now in the death phase. However, few remaining bacteria with virulence factor with them can actually survive in the extended death phase or the prolonged death phase. So bacteria with metachromatic granules will somehow utilize their food reserves. Bacteria with endospore can actually sporulate for them to survive much longer. And bacteria with flagella can travel and find other sources of nutrients. Okay, so how do we measure the microbial growth? We can do this by plate count. Okay, so you have here the original inoculum and then you perform serial dilution. And then after performing serial dilution, we actually placed only 0.1 ml of each serially diluted organism into the petri dish. And then we count the colonies. So after incubation period, you only choose between plates with 25 to 250 colonies, count it, and multiply it by the dilution factor. So for example, if 32 colonies are on the plate with 1:10,000 dilutions, you're able to count 32 colonies. You multiply 32 times 10,000, so it is now 320 CFU per ml, colony forming unit per ml. So that is the colony count. Or we can also do filtration. Okay, the bacteria in 100 ml of water were seed into the surface of the membrane filter, and then the filter is then allowed to be grown in the petri dish. Okay, it was so a filter such as shown here in the photo with the bacteria are much more widely spaced, so we can actually count them. Okay, and we placed it in a pad saturated with the liquid endo medium, which is selective for gram-negative bacteria. So the individual bacteria grew into visible colonies, and 124 colonies are visible in this example. So which means that there are 124 bacteria per 100 ml of water sample. And a more practical way of doing it is by doing the MPN index. MPN stands for the most probable number. There are 5 tubes, okay, in doing the MPN index. So for example, there are 5 tubes, okay, so combination of positive is 500. It means that the MPN index is 23. So if it's a 531, the MPN index is 110. Then we can also do the direct microscope count. In direct microscope count, we simply count the number of cells over the volume of area counted. So that would be possible if we're going to use the Petroff-Hauser counting chamber, because there are grid lines here when you look under the microscope, and we count the number of cells. So let's say there are 14 cells in the quadrant, so 14 over 8 times 10 to the 7 means that there are 17,500 bacteria per ml. So of course, a more accurate way of doing it is to measure the turbidity in spectrophotometer. So we can measure it in spectrophotometer. So by looking at the absorbance. So the more turbid, then the more bacterial growth there is. So that is an example of indirect method. So under the direct methods, we have the plate count, filtration, MPN index, dry microscopic count, and dry weight. For the indirect method, we have the turbidity, metabolic activity, and dry weight. And that's the end of our lesson for microbial growth. So wherever you are, always stay safe and continue to pray and build an altar, because healing is soon coming. Stay safe, everyone. God bless you.